Dr. Siawoosh Mohammadi is a group leader at the Institute of Systems Neuroscience, which is at the UKE in Hamburg. So he did a PhD in quantitative diffusion tensor imaging at University of Münster with Professor Michael Deppe, Howard Muller. And then he did a postdoc at the Department of System Neuroscience, where he is now. And his main focus is diffusion MRI, modeling and correction of artifacts, structural MRI, quantitative MRI for finding biomarkers, and also MRI reconstruction. So thank you very much, Siawoosh, for accepting to speak during our meeting. And yeah, looking forward to your talk.
Thank you. Do you hear me good? Is sound okay? Perfect. One important basically step where I was, you forgot to mention, I was at the Wellcome Trust for 4.5 years with Nick Weisskopf, and there the word in vivo histology kind of started to become part of my life. Okay. I would say scientifically it was also the best time of my life, so that's why I thought it should be mentioned as well. Okay, so but of course now it's also great to have our own group here in Hamburg. I'm sorry if I at sometimes have to think about it a bit, because I was still practicing the talk, so it changed it a bit from the last time, so kind of a bit might not be very fluent in it. So title of my talk is Towards in vivo histology using quantitative MRI application and validation strategies. So to say what we understand under in vivo histology, I would like to put two diagnostic tools in contrast. On the one hand, we have MRI, which of course is non-invasive. That's a strength. So we can put in a patient or healthy subject more than one time and measure him or her longitudinally without any harm. That allows us, for example, to look at different groups. Here in this example, we have Alzheimer patients, which were compared to healthy controls using diffusion tensor imaging, and they found changes along the corpus callosum in, I think it was the FA. However, the interpretation of these differences in the two groups they saw was not clear, so they couldn't really say what was the underlying cause of this change. And that's basically where MRI has its weakness at the moment. On the other side, we have ex vivo histology. Ex vivo histology is very invasive, so in order to look at it, typically you would take ex vivo tissue, so the subject has died already, or it's very via biopsy, so you remove a part of the brain, for example, in epilepsy patients or so, which is really invasive. And but then when you have the tissue, you can really look at it and understand the underlying mechanisms which lead, which was the source of these changes. Here, for example, in Alzheimer disease, you would have neurodegeneration, and these could have two popular or well-known sources. One would be demyelination, where the myelin sheet around the axons would become loose and vanish. And then the other one would be axonal degeneration, where single axons with their myelin sheet will degenerate. So here we have high specificity. We can disentangle different biological mechanisms from each other, whereas here we have non-invasiveness but no specificity. And the goal of in vivo histology is to get the best of both worlds. How do we do this? So in the ideal world, we would get our whole brain MRI image, we would do our group stats, but then we would have additional information. We could zoom into our voxel and disentangle different mechanisms, like demyelination from axonal degeneration, or say this is healthy tissue. Okay, this is the ideal world. Of course, we don't have the histology here, and that's where the biophysical models come in. So in this presentation, I'm not talking about all the physical challenges we have to face when we go to quantitative MRI, we increase the spatial resolution, we try to implement different physical models, but I will more focus on the biophysical challenges. So the biophysical challenge we have is that we have in a given MRI voxel, we have typically millions of axons, 100,000 of axons and other compartments. And what we want is we want to have one characteristic parameter that tells us something about the microstructure, which is the most important one. So basically what we want to get the most out of the MR signal that we can, but be as specific as possible with regards to this population of axons underlying it. And that's why we do what we can do best. We simplify. So we say there is one representative axon with a given myelin sheet, with a given diameter, inner and outer diameter, myelin sheet in blue, axon volume in yellow, and external areas. Then we would have something like a G ratio, which would be the ratio of inner and outer diameter, as you know. But still, as you can see, we have at least 3 different parameters we want to get from one MRI signal. And that's the point where we take advantage of another feature of MRI, which is that MRI has the possibility to get sensitive to different microstructural compartments. So in analogy to ex vivo histology, where we apply different stainings to highlight tissue compartments, we can do it under the microscope. We can do it also with MRI. By using different MRI, quantitative MRI techniques, we can basically make the sequence sensitive to different compartments. And the 3 contrast-generating microstructural features that we have would be iron, myelin, and the axonal features. And depending on the MRI contrast, we will be then sensitive, for example, more to the axonal features or more to the myelin. And here is where we basically are able to improve the conditioning, the conditioning of our inverse problem, because now we have more than only one measurement from the same microstructure by combining these contrasts. Okay, so to bring it now in a simplified, abstract way, the way from from our MRI measurement on the left to what we are at the end interested in vivo histology could be gone through quantitative MRI. So you have your MRI measurement, which is typically confounded by instrumental artifacts. You have different contrasts, which are also within an acquired image, so it can be T1 weighted and T2 weighted, and they have different contributions. By imposing imposing a physical model, you can now disentangle different quantitative parameters like T1 relaxation, T2 relaxation, and so on. But at this point, you are independent of the underlying or your physical model doesn't care about the microstructure that you have. It could be the microstructure of a banana, or it could be a microstructure of the brain, what you are measuring. What it will tell you is there are two compartments, or there is one compartment, and you have a T2 relaxation, which can be described by a mono-exponential, or it has a short and a fast decay. After imposing the biophysical model is where you translate your physical parameters into biology. And now basically your brain that you are measuring is composed of biological features. You again have a scale, a quantification, but it is now about axonal density, about myelin density, about diameters. So it's biology what you are showing now. Of course, that's a simplification, and there is also the possibility to go directly from the measured MRI signal to the biology. And that's what the talk is about. So I will talk about diffusion MRI, about multiparameter mapping on the MRI side. I will talk about diffusion tensor imaging, diffusion cortosis imaging, relaxometry, and MTSAT on the quantitative MRI side. And I will talk about axon diameters, and I will talk about G ratio and its constituents on the HMRI side. And here I will also link to the gold standard and try to validate what we are doing as far as possible. Okay, let's start with the axon diameters. That's kind of a quite huge project. So it's a collaboration with Leipzig, basically, where we have the Paul Flechsig Institute and the Max Planck Institute, where we get the histology data. We are doing some deep learning on our side, and we are measuring in Berlin using animal scanner with 9.4 Tesla, but I will talk about it later. Okay, the axon diameter that is measured with MRI is a bit different to what is typically reported in histology. So typically in histology, the old classical papers, they show, they report the mean axon diameter. And when you look at the frequency of these axon diameters, this is a typical shape, what you see. In MRI, however, typically an effective radius is measured. And if we take now the effective radius recently introduced by, or not introduced by, but used by the paper of Varad et al., for example, it would be a radius where this distribution is weighted towards the large axons. So the axons here at the tail, they have a huge impact on the diameter you have. So it doesn't matter so much what you measure here, but as long as this part is not correctly represented, you won't be able to use histology as a gold standard. And that's what this part of the talk will be about. So here now I come to the nice paper of Farad et al., where they showed that when calculating their arithmetic mean, they underestimate this MRI measure, and with the effective radius, they are close to it. And with their red data that they measured here, they kind of nicely were in the range of their measurements. Though I wouldn't say it's good enough to really pinpoint the single specific assumptions made in the paper, but it's already much better in terms of estimation than all the other works before, as far as I know. However, when they tried to translate this to his human data, that's where basically the weakness of the paper was. Because they didn't have ex vivo histology from humans, they used these classical papers like, these are, I think, the MRI data, but here you have now, no, here you have them, the old guys, Aboited, Community, and they measured with electron microscopy, counting manually the axons, and they had, I don't know, Aboited said per region, I think, 100 or 200 axons, and Community had a few more, but still not very much. So when they then calculated the effective radius here from histology, it was very different. So, and it's kind of by chance, because you have so little axons here, whether you will find the right measure, or you will, yeah, your estimation, your gold standard estimation will be okay or not. Because in MRI you will have 100,000 millions of axons, because these are even in vivo measurements, and they had huge voxel size. So they had 3 millimeter voxel size. So in 3 millimeter you will have millions of axons. So this is probably not representative what was measured here. And why that's a problem, I will show now using our data. So what we did, we measured human corpus callosum. We put it into a preclinical MRI system, where we went with the p-value from very low to up to 100,000. And tried to basically replicate their protocol that they suggest here to basically remove the signal from the extracellular space. And then we tried to estimate the ex vivo, the effective axon diameter from these measurements. I'm now looking first, I think, at electron microscopy data from the red regions here. And I'm starting with this one, S3. Sorry, this is light microscopy. That's a huge region of interest. And we did light microscopy and electron microscopy on the data. And this is kind of the gold standard in terms of histology, what was done until now. And then we wanted to basically see how good we can get with our deep learning based segmentations here. That's why we did both. But let's look first at the electron microscopy data. And when we estimate the electron microscopy based effective mean and compare it to MRI, we see that we are sometimes quite off. And I will show you why later. Now, even though we estimate, or we segmented, sorry, if I say we, it was Masha, who is the PhD student in Leipzig and who did all this amazing histology work and also the manual segmentation. She segmented all these axons. And here we're showing one of these distributions. And the arithmetic mean is estimated here to have basically the classical results. And then here you see basically how the weights of the effective mean would look like. And you see that huge part of this effective mean is actually determined by two axons, which might be there by chance or not. So if your field of view, which is in electron microscopy typically very small, is a little bit on a different place, you will miss maybe this big one, and then your effective mean will be totally different. So here shown what we have when we do light microscopy. We have much more axons, so we have several even MRI voxels. And we took one of these light microscopy images and we trained a UNet on it and then basically segmented the data and got then the axon diameters out of it. So the focus was here on the white area. So just for this one region to compare, we have 2 million axons in the light microscopy data, and we have about 1,000 axons in the electron microscopy where we did it manually. And now we think that with the light microscopy data, we have a much better representation of the large axons. And that's what we see when we look at the weights again. So we have much more axons here in the tail of the distribution. And when you look at the effective mean and you plot it, so that's the cumulative effective mean. So this, the end here would be the one which you measure if you include all the axons. But if you now remove the largest and calculate again the effective mean, you see that you get a smooth transition with light microscopy, indicating that you have a good representation of the large ones, whereas with electron microscopy you get these steps, which was what I said before. So if you by chance miss one of the big ones, you totally are off with this effective mean, which actually should be your gold standard. And that's what we get out when we do now light microscopy and compare it for several regions with MRI. Though I have to say the data are preliminary, so I wouldn't put my hand in fire for this data points here. This was a first shot. I think by now the results have changed a bit. Okay. So in summary, about the first part, electron microscopy might be a bad reference if you do it manually, and if you have this small field of views to validate MRI-based axon diameter estimation. Whereas this large-scale light microscopy images with deep learning might be the way to go for validation. And this might also help to solve some of the controversies at the moment on Twitter about these axon diameter measurements. Do you have any questions to the first part? Or do you want me to go on? Yeah, please.
I guess maybe a small one, but if it gets long, we can move it to the end there. Sorry if I missed it, but this has come up in our lab before. Maybe you could let us know how you define the axon radius in histology. Like how do you because it's not perfectly circular, how it's calculated or how it's defined?
I think in this with the results I presented here, we just use the equivalent circle. So assuming that it is, I think it's clear, right? Or should I explain it?
Like from using the area, you mean?
Yeah, exactly, the area,
okay.
and then assuming it's a circle.
Thanks.
But we can of course also use the smaller or bigger axis of an ellipsoid, and that will change the results slightly. I'm not sure whether we looked at it in detail, but of course you see that the diameters from histology become smaller. So though the correlation is still there, but you get a different slope. I'm not sure whether the slope changes, but that's exactly the things which we still need to do. So that's why it's preliminary.
Great, thanks.
And Siawoosh, this is really beautiful correlations you're showing. I was wondering, with the light microscopy, I realized that the limit is kind of 1.5 microns, really difficult to see anything smaller than that. But with electron microscopy, you should be able to go at higher resolution and hopefully report some of these sub-micrometer axons. Are you excluding them, not analyzing them at all? What's happening?
So I think in this analysis, Laureen, correct me if I'm wrong, we just took everything which was detected by the light microscopy, by the deep learning algorithm, but we are currently looking at this a bit more systematically. What happens if you replace the first part of your distribution with electron microscopy data, because basically taking the resolution limit of light microscopy and just replacing it by electron microscopy, or just skipping it totally and so on, and see how much of an effect it has on the effective radius. One thing that you need to keep in mind is this, I can go back, right? It is won't hurt the video. Is that the small axons have a very small weight on the effective radius. So even if you have a big error there, it won't be a huge problem on the effective radius at the end. Probably it's more important to really don't miss too many of the big ones. And especially if there is a huge one and you miss it, then it can be even a problem if it's a really big voxel size you have. But sometimes, of course, if you have a systematic bias here, it will also propagate through. And how much that is, that's exactly what we are looking at the moment in more detail.
Oh, thank you.
Okay.
Yeah, I just had a quick question. Yeah, I was very impressed also by the quality of your correlations. Just to clarify, I guess the take-home message here is that classical EM is a bad reference. You meant that what is a bad reference is a modality that uses like a very short like snapshot of the actual tissue, right? Because if we look at scanning EM, for example, you can get like as much FOV as what light microscopy offers, right?
Of course, that's why I called it classical. So what I mean with classical is basically the old aborted community works where they labeled them by hand and reported hundreds.
Right. So transmit EM.
Yeah, I think even there, the newer works that are around, which will probably come out soon, maybe make all this what we are doing unnecessary anymore. But that will be quite expensive if you have now 3D electron microscopy with a large field of view and you try to scan it all at this huge resolution. But of course, what you guys did, that would be also a good approach, I think, the scanning. Electron microscopy. Okay, so then I'm coming to the second part. That's more the G-ratio stuff, where you guys are much more familiar with than me, maybe. But I'll still do I will do the basics as well in some points. And if I bore you, just tell me, and I will just skip them. Okay, we start with myelin. With the myelin biomarker, so we use multiparameter mapping to get the relaxometry measures and MTSAT, and then estimated what we call here myelin density. So the multiparameter mapping would be this 3-point measurement with a contrast PD weighted, MT weighted, and T1 weighted, and calibration fields. From that, we calculate then the 4 parameters: proton density, apparent transverse relaxation rate, and longitudinal relaxation rate, as well as magnetization transfer saturation rate. And in order to now have the biophysical model that gives us the myelin density, we need to calibrate, and the way, how do I do it? Here. The calibration is typically done is we have this linear relations that we see often in histology between the gold standard, which would be from electron microscopy again, against the MRI biomarkers for different contrasts. This is a very nice work by West et al. You all probably know, where they had 3 different quantitative parameters: bound pool fraction, myelin water, calibrated already using literature values, and normalized water content, what would be the proton density in vivo. And you see that each of them has some offset and a slope. Since in vivo we are limited with respect to our gold standard histology data, we typically just estimate 1 calibration constant. And to do so, what we would do is we would basically take the linear relation and just reshuffle it so that we have an effective calibration constant. But this effective calibration constant, of course, is only then independent of the region in which we are measuring, of the offset, when the offset is negligible. If it is not negligible, then this calibration constant cannot really be generalized as it is used at the moment. I will come back to that later, showing some simulations from our review. But first I would like to go to the next biomarker, which would be then the axon density. And here we have now, again, different biophysical models we can use. That's not the historical way these biomarkers would be presented, but I went for simplicity. No, I got it, simplicity. So DTI would be the simplest, from which we can calculate using NODI DTI, the intracellular volume fraction, and then comes WMTI, which uses diffusion kurtosis imaging, and gives us again the axonal water fraction. And finally, we have the true NODI model, which directly then estimates the intracellular volume fractions from the diffusion data. One thing that is important, and which was nicely shown by the work from Nicola, I think, in terms of G-ratio mapping, at least the first time, is that with the axonal water fraction, you don't have what you actually would like to have, the yellow area here, the axonal volume fraction, because you dividing by the signal without the myelin compartment. And to correct for this, you typically have to have also a myelin measure, and only in combination from your diffusion measure, whatever it is now, and your myelin measure, you will be able to estimate the axonal volume fraction. I have to say this is one tissue model. And it might not be a full one, because I think what is neglected here is, for example, the fraction of unmyelinated axons. Okay, now that we have these two measures, axonal volume fraction and myelin volume fraction, we can go to the G-ratio. This I think I can really skip. What the G-ratio is, we had it before, and why it is important as well, and how it is calculated. I'm citing you guys anyway. But then I'm reporting here our study where we basically acquired the two-shell MRI data, diffusion data, and the multiparameter mapping data, so that we had two biomarkers for myelin and two for diffusion, and could calculate different G-ratio measures. So we used these two, and we had two from the diffusion data, which are shown now here. So we had the magnetization transfer saturation rate, the proton density, from which we then calculated macromolecular tissue volume. And then we had on the other side NODI and tracked fiber density, which was a measure which we used the first time when doing G-ratio mapping, which could be extracted from one shell data as well. And then here we looked basically at scan-rescan reliability, and we found that this combination NODI and MT-SAT was most reproducible. However, this doesn't tell us anything about the accuracy. It's only the precision which was looked at here. Okay, that's what I said. And now to look at the accuracy, I would like to come back to the calibration again. So we had these different markers which have different offsets and so on. And here, for example, the offset is very small. This is not really translatable to the offset. So, but there is another paper by Berman, so the group of Aviv Mezer, where they used the data from West et al. And calculated the MTV from, and that's what we used for our simulations later. So the simulations that we did here went the following. So we took the data from Mark Doe's, or we got the data from Mark Doe's, which were published here in this Clem paper and in the West et al. Paper. And we looked at so they had the histology and the diffusion data, and we were basically interested in generating ground truth G-ratio maps, and then use basically these relations to see what is going wrong when we are doing the single point calibration, which I talked about before. I will show you in a minute what I mean by this. So, but first, what we needed is we needed the histological relation between in these mice models. Now we have 4 mice models, sorry, 3 mice models and a set of controls. 2 of the mice models were hypomyelinated, the Richter and the TSC2. So the TSC2 were severely hypomyelinated, meaning that there were almost no myelin, and the P10 were hypermyelinated, so there was more myelin than in controls. And then what we looked at here is there a heuristic relation between the myelin volume fraction from histology and the axonal volume fraction from histology, from electron microscopy? So that we can calculate now a ground truth G-ratio using this relation, which we fitted, to then take this ground truth G-ratio and compare it to what we get from MRI. So, and how did we calculate the MRI G-ratio? For the MRI G-ratio, we kept the axonal volume fraction the same. So we don't care about, in this simulation here, since we don't do any calibration anyway for the diffusion data, we don't care about the axonal volume fraction. We assume that we have a perfect measurement for axonal volume fraction, and we are only interested in what happens if we now have a myelin volume fraction measured with MRI, making the errors we showed before. So we have an offset which is not accounted for when we do the calibration, the single point calibration. And that's basically the results you get. So here we always plot our G-ratio from MRI with the ground truth, and we do 3 different things with it. One, we don't do any calibration. We just use the measured MRI G-ratio and plot it against the ground truth. We use the single point calibration, taking as a reference measure the G-ratio, or taking as a reference measure the myelin volume fraction. The myelin volume fraction is very simple. So here you would have on the left side the histological myelin volume fraction in the region of interest, and then divided by the MRI measure in the region of interest. From that you get your effective constant, and then you just multiply your MRI with this effective constant, and that would be the myelin volume calibration. And you see that there is no big difference to the G-ratio calibration in terms of the results. How that's done is a bit more complicated, but I think also not important for now. If you want to know the details, you can just look in our review. But what is important is the results here. So what you see is, first of all, the offset plays a big role. So the bound pool fraction, for example, which had a big offset, so let me go back. Bound pool fraction has a big offset as compared to its slope, is quite off from the ground truth G-ratio. Even if you calibrate using the single point calibration, it's still quite off. And the other two, they don't get also necessarily much better in terms of their error, at least, when you do the calibration. They get better in terms of the offset, but not in terms of the error. So, for these examples I showed, the single point calibration seems to be insufficient because they had a non-zero offset. So and sometimes it seems to be better to do no calibration. And I think, for example, that's also what Aviv Meser's papers, where they use the MTV as a biomarker did. However, I have to really make clear that what we do here were based on ex vivo data. So I don't expect that the in vivo measures will have this huge offsets. And the correlation between to the myelin measure might be totally different. So it's not translated. So I wouldn't basically judge from our simulations about which MRI marker is a better one, even though the bound pool fraction here looks very bad, but it is because it's ex vivo data. So I don't think that this correlation we would see also in vivo. And in fact, we just looked at, in this review, we also looked at how our MTSAT correlates with the MTV measure. And we compared this to this data here, and it looks totally different. So there you already see that the interrelation between the two is not the same as so between in vivo and ex vivo data. So you can't really do the extrapolation, but I think so conceptually it's important to know that we need to think about a better calibration approach. So single point calibration seems to be a problem here, or at least we need to be aware of it. Okay, now let me summarize. You remember this image from the beginning where we said what we want to do with in vivo histology. We want to zoom in and we want to disentangle different neurodegenerative diseases by the information we have from MRI. And the simple G-ratio model can already give us the information which we otherwise would get from electron microscopy if it is calibrated correctly, of course. Because a demyelination disease would only affect the myelin sheet, so the blue area, but would leave the yellow area, the axonal compartment, the same, whereas axonal degeneration would affect both. So having the G-ratio and its constituents, we can already distinguish these two mechanisms from each other. And that already shows us the strength, the potential of these biophysical models after they have been validated, of course, also in pathology. In our group, we are trying to basically develop these pipelines for validation. And when these grants at some point will be at their end, I hope we can also publish them and provide them as open data so that other people can make use of this. Because I think this was really a strength, for example, with the data that we got from Mark Dose, which helped us a lot to understand the G-ratio problem a bit better. And I hope we can do something similar also with this axon diameter data at some point when we have the data and the tools ready, so that maybe other people come up with a better model than which is there at the moment. Okay, finally, I would like to thank people in my group, of course. So the Thibault guy, you know from before, and now basically he's gone, unfortunately. Also others are gone and new people have come. We have great collaborations with mathematicians and of course the MR physics guys, Nick and Martina in London, Thoralf Niendorf in Berlin, and his co-workers, the postmortem people and the histology people. And finally, I would like to thank my funding agencies. And thank you for your attention, of course. And sorry if I was too long. I don't know. What's the time?
It was perfect.
Okay.
Thank you, Siawoosh.
Thank you.
Stop the recording.